Hello, everybody. Welcome back to Coffee with EMBL. So if you're enjoying some coffee, I've got my cup of coffee with my official EMBL mug, and I plan to enjoy it. We've got a fantastic program again, and it's just wonderful to see again people coming back to this. This has been going on now for 8 weeks. My hair is 8 weeks longer, and I don't know whether I'm ever going to get it cut. And the first thing I wanted to say to everyone was Happy World Environment Day. So June the 5th is the celebration of World Environment Day. This was established back in 1974 by the United Nations and is celebrated every 5th of June. So it's a happy coincidence that today is World Environment Day. And the aim of the United Nations was to highlight some of the really, truly global challenges that we face. And the main theme this year that the UN was highlighting was biodiversity, and indeed the loss of biodiversity as a major topic. But of course, they also recognize amongst these global challenges, the challenges of climate change, increasing pollution, and very relevant to our discussion series, the spread of infectious disease. So these are major challenges that really need to be addressed at a global level. And I think it's also very apposite for the future of EMBL, who have chosen to embrace these global challenges. And the new EMBL director, Edith Herd, has led a focus for the future for EMBL in its future strategic forum moving forward, to plan to bring a new molecules to ecosystems program. And this whole theme of biodiversity and environmental challenges will become, I think, a big part of the future of EMBL's future research. And we're going to hear a lot more about that on our EMBL World Alumni Day, which is scheduled for July the 17th. And so these are some of the themes that we will be talking about there. Particularly the idea of planetary biology and environmental research. Anyway, happy World Environment Day, and I hope we can recognize these challenges without getting too depressed about them. Obviously, the theme for our Coffee with EMBL sessions for the last few months has been the global challenge we're all in the middle of, which is the ongoing coronavirus pandemic. And that will also be the theme of today's speakers. We're going to be hearing from Michael Levitt from the University of Stanford Medical School. And Michael, who is, as I'm sure all of you know, was the Nobel Prize winner in chemistry in 2013 and is a data science expert who's been recently looking in detail at data models for viral epidemics and disease progression. And I'm really excited to hear what he can share with us about understanding data and how that relates to our understanding of the data around the pandemic. We will also be hearing from Ennio and from Ishaan in Italy and in India. And we're going to be talking about the landscape for vaccine development. Ennio is a GSK director in Siena, and he's going to be talking about the whole landscape and systems for developing vaccines, which is highly relevant to our understanding of the progression of the epidemic. And Ishaan is someone working on COVID-19, one of the EMBL alumni who's directly working in this area, who will share with us some of his thoughts on research into COVID-19 and also the future in the post-COVID world. So that's what we've got planned today. Of course, we also want to make this as interactive as possible. We planned, as in recent events, to have a EMBL Coffee Second Cup for anyone who's able to stay on. So if you don't mind, maybe just before we start, I could just ask one question to anyone in the audience who is currently actively employed, whether as a scientist or in some other capacity. We were interested to understand throughout the world and throughout the participants whether it's been possible for you to return to work yet. So if you have your participants tab open and you see at the bottom you've got the option to click on the yes or no tabs. I'd just like to ask you the question, have you been able to return properly I'm not talking about working at home, I'm talking about going back to your place of work. So I'm going to vote now. I'm not able to go back, so I'm working, but I'm working from home, so I'm going to vote no. Okay, so I see that the overall response is pretty mixed, but still just over half of the people who've replied have not been able to go back to work yet. That's interesting. That's quite remarkable, actually, that as we're well into the third month of this lockdown, that so many people, including many people who I think can contribute a lot by being back at work, have not been able to return. There's a general point I think will be coming up very shortly, and I think it's something that will emerge naturally from the discussion we'll have with Michael and from his presentation. But I must say something I personally have been struck by. It has been the difficulty in science effectively communicating. Science inevitably is a community where it's right and proper for there to be disagreements, differences of opinions, arguments, debates as we attempt to come to terms with emerging sets of data, and then gradually develop a synthesis that most people can agree on. And I think it's been quite a challenge to see how science is being portrayed, both in the media and how a subset of science is perhaps disproportionately affecting the actions of government. I have to say that the success and the interest I've had from these events over the last 2 months have led me to feel that there could be a very valuable role for the EMBL Alumni Association as a truly global collection of intelligent, highly motivated and relevant people who can help synthesize opinions in a more informed way than sometimes happens in more narrow national debates or subnational debates. So I just wanted to ask the question, do you feel that developing future communication fora like this and activities for the EMBL alumni community would be a valuable way for science to improve society and its ability to communicate with global media and with our governments? And could vote yes or no. I'm going to vote yes. Okay, well, so again, from the people responding, there seems to be a general consensus in favor of that. And that's something we'll come back to and think about in the future. Again, as always, please do add comments to the chat. And send us your comments on what you think we should be doing as an association. In terms of the association, I'd like to just kick off today by welcoming Fatima to say hello, who is the leader of the Alumni Association.
Hello, everyone. It's a pleasure to greet you all and happy Environmental Day. I think this is an example. The cafe with EMBL is an example on how, a disgrace that has been this pandemic can also be turned into opportunities. And I think we actually are learning a lot on, communications. And with these things, we are even, I think, feeling more joined, bound, and together than if we could meet physically. So I think this is something to actually exploit. We keep having, even if after the pandemic is over. You know that the Alumni Association is an evolving creature. We have a big renovation of the board recently. We have a lot of ideas and a lot of excitement on trying to connect alumni and to trying to respond to alumni needs. And, I think I will inform you more specifically after our next board meeting, which is next month at the Alumni Day in July. And in the meantime, please keep safe. And as the time is running, I lead the mic to Angus.
Thank you very much, Fatima. We'll look forward to it. And again, I really just to emphasize how global the EMBL Alumni Association is. And this is our fourth coffee with EMBL. Again, we have about 150 people registered from countries all across Europe. but also many people from the USA and also from Australia, Canada, Ethiopia, Lebanon, Russia, Saudi Arabia. So we've been typically sampling scientists from more than 30 countries and 5 continents. And I think that's a fantastic opportunity to really understand a diversity of views at a global level. So without further ado, I'd like to introduce our first speaker today, Michael Levitt. Hello. I'd like to welcome you to Coffee with EMBL. Perhaps you could just start off by just saying a few words about yourself and your association in the past with EMBL, and then tell us what you've been doing to analyze data during the coronavirus epidemic.
Okay, so when I get to talk, I'm actually going to be sharing my screen. But I never really was at EMBL other than being part of the SAB in the 80s. I see that 2 of my EMBL friends, Arthur Lesk and Chris Sand, are both part of this meeting now. I admitted to them. As I said to you before, there's been a real lack of global discussion about this. This is a very difficult issue. The decisions are all very complicated, and for whatever reason, there seemed to be a desire to have as little discussion and advice as possible. So I really am happy that EMBL has changed this. Let me share my screen now. So basically, my first observation is that this is not really an observation. This is just an example of the reports that I was putting out from the 2nd of February to the 3rd of March. This was the first one. And what one actually sees here is that what I call the fractional change, an index much easier than the R value, just the ratio of today divided by yesterday, actually indicated quite early that the epidemic in China was coming to an end. I had no basis for doing this, but I got involved. This report got leaked, translated into Chinese, and as a result, I was hooked. And let me just sort of now give you what are my 3 observations. So one observation is that this virus really does look the same everywhere. A lot of people have said to me, how can you believe any data coming from China? And I think this is a pretty interesting illustration. So in China, the outbreak in what I call China non-Hubei, which is China outside the province of Hubei, this black line shows how the number of cases increased on a log scale. The red line is the number of new cases on this linear scale, and the blue line is just the gradient of the log plot. So what you see is that this is a familiar flattening curve. The cases peaked, and this is the exponential growth rate. The exponential growth rate was decreasing from the very, very first case mentioned. Now, you might say, well, gee, but if now you go forward, I guess 2 months, from 15th of January to the 13th of March, you discover that in New Zealand, the curves are exactly the same. The dark line goes up, which is the number of cases. The red line is the number of new cases. And the blue line is the deceleration or the exponential growth rate of cases. So one thing I have seen by looking at all this data is that in general, with data, you believe it until proved otherwise. There has been a problem with the data, but the problem has been very general. And I think that the Chinese data, in fact, was surprisingly accurate. One thing that's good about the Chinese data is they don't really have weekends. So we see this phenomenon in much of the world where deaths and cases decrease dramatically on Saturday and Sunday because they aren't being recorded. Anyway, this is my first observation. This is a virus that doesn't grow exponentially from day one. So anything that assumes expansion on the scale, exponential growth would be a line like this. What this is saying is that your bank pays you 100% interest the first day, 50% the second day, 25% the third day, and in fact, the interest rate is decreasing exponentially. Okay. Let me now move to something different. Once we saw that this thing was growing slowly, it immediately became very important to try to estimate what would be the total level of the catastrophe. And go here first. And this I did by looking at the Diamond Princess. This was essentially what one can do is one can say the Diamond Princess, which was a cruise ship, had a population of 3,700. And on that cruise ship, 20% got infected and 7 died. People have died subsequently, but those are the numbers that you see. You can imagine this as being a closed population. A cruise ship is not a great place to lock down. The population density is 40 times higher than Hong Kong, 250,000 people. per square kilometer, very high density, shared air conditioning, and so on. And yet, even though the population was very old, only 7 died. So you can then take this and extrapolate to other areas. So, for example, if you start out by taking numbers of deaths from Neil Ferguson and others, you discover that if you apply these same numbers by just proportionality, this is for the UK, 500,000, around 2 million for the United States, and that would have given you on the Diamond Princess 58, but there were only 7. So now you can just divide everything by this factor. The UK drops down from 500,000 to 61,000. The United States drops down to 260,000, and the Diamond Princess is 7. Now, this is a level where you have 25% infection. Basically, everyone who can be infected is infected. The UK is much closer to getting to this point now with its current death. The USA is about half the way there. So this gave one a kind of simple measure of what the level of infection could be. Again, this was very, very simple. And it was an interesting check-lane thing to the city of Wuhan in Hubei in China. In Wuhan, there were actually a lot of deaths. Basically, in Wuhan, there was something like 2,600 deaths, which was then corrected to 3,600 deaths when they added more deaths. If you applied the numbers that were given by Ferguson et al. To Wuhan, you would have expected 30,000 deaths. When you apply the Diamond Princess numbers to Wuhan, you actually get 3,600 deaths. So these two completely independent places seem to saturate at about the same level. So now let me just move on to my third point, and that is something which is actually very European. There's a European website called Euromomo. Very surprisingly, they don't even own the Twitter. Some person is Euromomo on Twitter. And this basically shows you, this is a database that collects data from a population of about 350 million. And what we plot is every single week, what is the number of excess deaths? In other words, what is the total number of deaths in this area? We can see that now clearly in the next picture. What you see here is, this is time. This is 2016, 17, 18, 19, and so on. And what you see over here is that I'll move this away. In 2020, there's this big peak that comes from COVID-19. Now, we don't know it's from COVID. This is just the total number of deaths from COVID. And we have a baseline, which goes up and down. It's like the seasonally adjusted baseline. It's higher in the winter and lower in the summer. So basically, the total deaths, which are shown here, are above this baseline, and the amount that are above the baseline is what we see here. And from this, we basically see that in currently up till, I think, yesterday evening, the numbers were released on Thursdays, there were 170,000 COVID-related deaths or deaths in the COVID season. And that corresponds to 133,000 deaths in the most recent severe flu season. It's important to note that there was no flu in 2019 hardly, and really nothing in 2020. So basically, we can put those numbers together in this table and discover that overall, COVID-19, one thing I should also have said, this peak is gone down. And this peak is now actually below the baseline. So we're not likely to see any new COVID-19 deaths. There may well be a kind of dropping below the baseline here as people who would have died later actually died during COVID. Now, the final interesting thing is from these numbers is we can do this calculation using Euromomo data for different age groups. And what you find is that for COVID, about half the people who have died are over 85, and only 8% are younger than 65. And I was amazed to see that if we look at exactly the same data for the 2017-18 flu season, we see again 8% being less than 65, here a little bit more than 85, and we see that these two totally different diseases, COVID and influenza, have about similar mortalities and similar distribution of mortality with age group. One other thing you can do is you can actually look at how the baseline varies, i.e., you can go here and say, what is the baseline for 65-year-olds? What is the baseline for 85-year-olds? And this will give you the so-called natural death. Now, I guess there's never such a thing as a natural death. We all die of something. But what you find, to my surprise, is that natural deaths, which also include accidents, are actually 16% for people younger than 65 and only 42% people over 85. So young people are half as likely, proportional, to die of COVID than they are of everything else. And young people are not likely to die very much of everything else. So that is basically what I wanted to tell you. I've gone through it probably too quickly, and I'd love to have questions.
Thank you very much, Michael. That's a great overview. I appreciate it as well, because I know from the YouTube videos you posted and the other things online, I highly recommend to everyone listening to go and have a look at Michael's discussions of many of these points on YouTube and on possibly on Twitter. Perhaps you comment if that's also, you think that was your preferred medium.
Yeah, well, I forgot to say that. Let me just share my screen again. And this requires me to apologize. My Twitter handle is horribly pretentious. It was all designed just to complain about airlines and banks. And then in the middle of March, I discovered that I needed data for deaths in New York. So I asked Twitter as a random first tweet. And to my amazement, I actually asked the mayor's office in New York, got no reply, but about 20 people anonymously sent me all the death figures. They were obviously listening in. And I suddenly realized that as a way of data retrieval, Twitter is quite amazing. So I actually find the discussion on Twitter, maybe the discussion here will be as good, but I haven't found any forum that gets even close to Twitter in terms of discussion. I'm going to stop sharing again. Okay, I'm done.
Okay, and just to follow up on the comment that Michael made earlier to me, was that he was looking for more critical feedback. So I said I'd invite everyone here to give Michael as much critical input as possible, because like he revels in interactive critical feedback. But one of the things that you mentioned there, I thought was extremely interesting, was the data from the cruise ship, where you had this super high density population. You had a population that was biased towards a more at-risk population with a lot of older than average people, and yet the statistics were what they were. Now, recently, Carol Friston, who's also not an epidemiologist, but it has expertise in data science, we discussed this earlier, from Imperial College in London, has carried out some parallel analysis to to Neil Ferguson and other epidemiologists, where he has argued that looking at the data retrospectively, he's proposing that there is something, I think it's starting to be called dark matter of immunology, where he's arguing that in fact the population in each of the European countries or in any of the countries is not uniform in its susceptibility to the virus. And he has argued that there is actually, for reasons still unknown, perhaps exposure to a different coronavirus that's cross-protecting or some other reason, that a significant proportion, anything from 50 to 80% of the population simply are not at risk from this current virus. And that's a very provocative and very interesting comment, because if that's true, it has quite a lot of implications for social distancing and lockdown and how society reacts. Are you familiar with this, Michael? Do you have any comment on whether we can infer whether there's as many as 80% of us who are not sensitive to it?
What is interesting is that the little part of my talk where I talked about work that had been done in England, I actually did raise this with Neil Ferguson between the 18th and 22nd of March. I didn't get very far. I said 7 people progressed various things, and he would have said 500,000 people in England and so on. So basically, I actually said something which I hate saying. I basically said whatever. I didn't get into it. I think that it's absolutely clear that if you look at various areas around the world, whether it's Iraq or Italy or Wuhan or New York City or London, they all seem to end with a mortality of a little bit less than 0.1%. This is not, I'm not talking here anything about infection ratio or whatever. I'm just simply saying where this thing seems to stop. The average for all of Europe is 480 per million population. New York City is worse. It's more like 1,200 per million population. But this seems to be an ending point. Now, whether this is due to some kind of herd immunity, whether it's due to some kind of resistance, there are many explanations for it. I mean, I just honestly feel that we don't know yet. But I think it would be very, very, it's like a runaway truck that just stops at a certain point. And this is how this virus is behaving. It's also surprising, and something I don't understand is, why does the exponential growth rate decrease exponentially from day one? And it is actually true everywhere. It's true in Sweden, it's true in the United Kingdom. The one thing that I got to trend on Twitter is Gompertz, the name of a British 1525, so it's 200 years ago. And the Gompertz function, which seems to fit this epidemic perfectly, comes back then. And it's a function which describes a situation of growing under restraint. When bacteria grow in this food, they grow with a Gompertz function. So from the very first moment, the fact that the exponential is decreasing linearly on a log plot says it's Gompertz. And this, I think, is very, very significant. The information was there. I actually missed it until the middle of March. So I didn't actually see this. I used the sigmoid function to fit things. But the fact is that it's Gompertz. And this is telling you that the virus is in some way self-limiting. It's either self-limiting because it can't find people to infect. It could be self-limiting because there are many silent cases that are also infectious who are infecting people before you get to them. So we don't know. But I think all of these things are coming together, and we need somebody to explain. I mean, the key experiment really was Sweden. The group at Imperial predicted between 60 and 90,000 deaths in Sweden. Sweden, which everyone is now saying is having a really hard time, will end up having, I would imagine, around 500 deaths per million, which is exactly the same as all of Europe. What Sweden is now doing, which is very clever, they're actually spreading the disease from Stockholm to Skåne, the area of Lund, and to Gothenburg to try to get the whole country saturated. So they are obviously being very, very clever about this. The average European number million could be because some areas have been hit, other areas have not been hit. So I think if your country has been uniformly hit, you're actually in a much better place than if only half your country's been hit.
Thank you. So I'm just seeing that there's a few questions coming in. So I'll, if you'll allow me, I'll just read these out. The first question from Meltzer is: do you think if coronavirus is mimicking the seasonal effect that we see typically in a winter from influenza virus, does this suggest that there will be a seasonal COVID infection cycle in future as well? I hope I've got the sentiment of the question correctly.
So I think that what is important to realize, and it's obviously a hard thing to say, but in every society there are people who are closer to death than other people. And you can imagine this is a forest with some trees that are kind of getting ready to be blown down. And because there's been such a weak flu season, both in the winter of 1920 and also in the winter of 1819, you could argue that there were people who were on the edge. There were actually a large number of excess people, the people who would have normally passed away during the missing flu attacks were there. So I think there's definitely something of that. The European numbers actually are completely mirrored in the United States as well. The data isn't so clear now. So I think there could be something like that. It's not clear. I think people need to remember that the flu virus is a very special virus because it mutates. Polio doesn't mutate. The other coronaviruses we have in common cold don't mutate. I am not sure that we're going to get a new variant of coronavirus coming back. In fact, coronaviruses, when they do mutate, become less and less, generally less and less virulent. So I think the indication from the data is that if it comes back next winter, you get colds in winter. The reason you get colds in winter is very simple. Cold viruses, these viruses don't like being in dry heat. So if outside is dry heat, virus is not going to be easily transmitted. Wet heat, equatorial Africa has endemic influenza all the time. So there's an issue here. But I think if it comes back next winter, I think it'll come back mostly in those places who escaped the first time, because they'll either have the weak trees still standing, or they will not have had the level of immunity that we might be getting. I don't think it's going to be anything like a serious second wave. A lot of people have compared in a state of great panic COVID with the 1918 influenza, and it isn't even like the 2009 H1N1. It's a relative, it's being 30% higher than 1817 influenza is not a terribly big indictment.
So we have a question also from Olivia. I'd like to shout out to Olivia, who kindly has agreed to speak at our next Coffee with EMBL session in 2 weeks' time. And Olivia was asking Michael, how do you explain super shedder events, such as the choir in Washington State, where one person infected almost the whole choir in one 2-hour practice?
So I think what seems to be the case about this virus is that it's really spread. If you want to make a very simple rule, instead of saying the 2-meter rule, and I think if you look at somebody and talk to them with an uncovered mouth, then micro droplets of saliva from you are being inhaled by them, and therefore this seems to be the mechanism. People are often infectious before they even know they're infectious. So a choir, a football game, or fitness, one person singing loud will fill the air with viruses. People will be completely unaware of any protection. And, the corollary of this is, how come there has been no other outbreaks in all the Asian countries that wear face masks? Japan should have been another Italy. By its demographics, Japan has an even older population than Italy. It has very few children. It's a very aging population. Japan has never eradicated coronavirus. It's just there all the time, but they've never really had a super spreading event. So I think if we had realized very early on to wear face masks, which a lot of people don't want to do, but the economic consequences of wearing a face mask is immeasurably better than the economic consequences of a lockdown, it would have been a great idea. Yeah, and it doesn't have to be an N95, a handkerchief, or even, but if I was speaking to somebody in front of me and I spoke like this, my hand would be covered with viruses, but it wouldn't go to them. So I think there were some simple things we could have done. People didn't do this. I'm kind of shocked by plus there was a lot of hostility. I mean, some of the people listening will know that in very learned organizations, people got very upset with me if I dared suggest that we should be wearing masks. And this was in the middle of March. So, we really have a lot to blame ourselves for.
So there's a lot of interesting comments coming up. I think general comments for us all to discuss. I think I tried to come back to some of those in our discussion after the speakers have come in. Just one comment that's coming up in this comparison of COVID and influenza. One of the bits of information that I've come across since our last Coffee with EMBL is that we are often talking and comparing COVID with what's recognized as the last great global pandemic, which was apologies to anyone from Spain, but it's often called the Spanish flu, although it didn't come from Spain, of 1918, which killed more than 1 million people worldwide. But actually, apparently the pandemic before that killed 1 million people or more worldwide was called the Russian flu of 1891. And a very interesting analysis of the clinical symptoms of the so-called Russian flu of 1991 actually was identified by a group in Belgium in 2004, I think, as actually looking almost identical to what we now see with COVID-19, or very, very similar. So what was called a flu outbreak, because it wasn't, there was no PCR or DNA testing, obviously, back in the 1890s, but it may actually have been that the great pandemic of 1890, called the Russian flu, may actually have been very, very similar to COVID-19. And it's thought that what has happened is that has turned into one of the seasonal viruses that now causes colds, but kills very, very few people. So we may actually be in a position of seeing a pandemic which will evolve or change or dissipate into being another type of cold virus, which never goes away, but isn't the massive health problem it is today. But before that happens, I'm not suggesting that's going to happen if at all, within the next few months. And many people are placing their faith in the development of a vaccine and getting us out of this. I believe Bill Gates and others are devoting and putting a large amount of money into trying to help to do this. So I'd like to welcome Ennio from GSK in Siena to be our next speaker, who very kindly has agreed to give not a GSK perspective, but a global perspective on the vaccine development landscape. So Ennio, may I hand over to you, please?
Thank you. So my name is Ennio Gregorio. I'm head of the Research and Development Center here for GSK Vaccine. So I'm a vaccine expert. I was in EMBL back in 1996 up to 2000 in the gene expression program with Matthias Hentze. I was working on RNA at the time. Then I moved to innate immunity in Drosophila, toll-like receptor with rural MH. And then I started to work on adjuvants. I joined the industrial sector working on vaccine adjuvants. And then I made my career in the industrial sector. And now I'm heading the vaccine research and development here. So just to link to previous talk, I think that thanks to the speaker, thanks to Michael Levitt, because the way governments present the data, epidemiological data, is always linked to the lockdown. So everything is shown to us like this is the effect of the lockdown. Of course, they have to do it because the lockdown was painful and induced a lot of pain in people and the economy. But I agree that indeed the effect, epidemiological effect that we see is a combination of effect. For sure, there are people that are not susceptible because they have cross-reactive immunity with coronavirus, may not express the receptor of the virus, or maybe they are very good interferon responders. So there may be several reasons. And there is a seasonality. I think that the lockdown had an effect. The data to support that is that in Italy, for instance, in some regions, you have very, very high numbers, like in Lombardy. And what the lockdown did was, I really think, prevented the spread of the virus in the south of Italy, for instance. So what I believe is that the risk of a second phase in winter is there. I hope it's not true, but I think it is there because there are some region in the world, even within Italy, that have many, many cases. It was the first country to start, where not everyone has been exposed to the virus. That's what I believe, but we will see. In any case, everybody is waiting for the vaccine to prevent this second wave to come. Well, this will be very difficult because we want to have a vaccine to prevent second wave. We will need a vaccination campaign in September. And I think this is probably very difficult to achieve. Just to give you an idea, vaccine research and development takes between 10 and 20 years. Those are the timelines for not standard timelines of vaccine development. It takes 2 to 5 years in preclinical discovery, 6 to 10 years in clinical development, and preparing the site approved by EMA, FDA for manufacturing takes several years. So what we are trying to do here is really unprecedented. The timelines, existing timelines for COVID vaccine development are not standard timelines. And the 4 main reasons why vaccine producers believe that they can accelerate are the following. First, the knowledge of previous coronavirus. We went straight to the target. Spike protein, pre-fusion construct were rapidly available. The second one is the use of new platforms, essentially RNA and viral vectors. These platforms don't have antigen-specific process manufacturing. They are really standardized process manufacturing, preset. If you change the antigen in adenovectors or any vector or in an mRNA vaccine, the production process is always the same. So it's very fast to get preclinical candidate and even GMP clinical candidate. That's why it took a little bit more than 40 days to Moderna to go from the design of the construct to clinical trial material. It's unprecedented. Of course, there is an unprecedented financial investment, private and public, so a lot of resources are mobilized. But there is also an emergency-based approach from regulatory authorities. Vaccines are given to millions of healthy individuals. So health authorities have put the bar, especially for safety, very, very high. This time, there is a less risk-adverse approach. The clinical package was pretty minimal. Even toxicology package, I haven't seen it because I haven't seen what the competitors have done. But if I look at the timelines, it's impossible to build the standard toxicology packages. So it was very fast to get to phase 1. And with novel technologies like RNA, more difficult to get to phase 1 with standard technology that have, by the way, highest probability of success because they are standardized. There are 10 vaccines in clinical development, more advanced viral vectors and RNA. Very soon followers are recombinant protein adjuvanted. They have to be adjuvanted, and we can discuss that if you want to have a discussion on that. Inactivated virus, live attenuated virus, Pasteur, all type technologies are also coming, and preclinical data are very good. But those things are difficult to make in a standardized way, inactivated virus and live attenuated virus. Well, and I just end with this part. What are the major future challenges for vaccines? First, demonstrate efficacy of vaccine in a context of declining symptomatic cases. How can we assess if the vaccine is effective or not if we only have a few hundred cases, most of them asymptomatic? There is a risk to have PCR positive as readout because the vaccine may actually prevent disease but not infection. This is actually known for many vaccines, including flu vaccines. So it will be difficult to demonstrate efficacy. Exclude vaccine-induced disease enhancement will also be almost impossible because the number of cases will be so low that how can we assess if vaccine makes even worse on 1,000 vaccine subjects if we just have 1 or 2 symptomatic case in my vaccinated group compared to placebo. Scale up production to meet global vaccination need is a big issue, especially for novel technologies like mRNA and viral vectors. They don't have any commercial vaccine available. So everything needs to be done. Nucleotides, in vitro transcription enzymes, the release assay, everything is new for RNA vaccine. At the commercial scale, has never been done. So there may be risk there, and we don't know how many doses we will be able to produce. Demonstrate persistency of protection, probably less important, but I think it's also important. COVID mutations, there are some that may induce evade immune evasion, probably not, but it's another risk. But the main challenge is time. We may have the vaccine when it's too late. And this is even more clear after the previous talk. We don't even know if there will be another wave. And I have to say, my experience with the vaccine in response to infection has always been a lot of noise. Everybody wanted us to make vaccines, and a lot of resources were mobilized. But the vaccines, in my experience, were always available after emerging infection, when emerging infection was gone. Now, we all hope that it's going to be the case because that will mean that COVID will be gone in 1 year. But this is my really short summary of the very complex situation. Imagine 10 vaccines in a clinical trial, 123, but the update in June 2 in preclinical. So there are 133 vaccine at least where people are working for COVID. It's a tremendous effort, a lot of resources in the science. Thank you.
Thank you very much. I'm sure people have a lot of questions, but perhaps just to start it off again, I've not been super optimistic that a vaccine is going to arrive to get us out of the lockdown and out of the current crisis. So I'd like to have a vote with people to see what the general feeling is around the community. There's obviously a vast amount of money and resource being poured in to vaccine development, even though I think I'm right in saying, please correct me, Ennio, that previously there were no terribly effective vaccines ever developed for coronaviruses or for SARS or for something else. So I'd just like to see the opinion of people who'd like to vote yes or no under the participant key. Do you think a vaccine will arrive to help us escape from the coronavirus epidemic? So can you comment any further on that?
Yes.
In fact, that's what. Yeah.
I believe that, all the vaccines in the pipeline are based on one assumption. And the assumption is that by inducing neutralizing antibody against spike protein, so antibody that neutralize the binding to ACE2 receptor, we will prevent the disease. That's the assumption. Maybe if there is no correlate to protection, it may be true or not. I believe it's true. I believe that the vaccine that induces high level of neutralizing antibody very likely will be able to at least reduce the disease of people that are infected with COVID. These vaccines, after saying that, these vaccines also have the potential to induce, most of them also CD4 response, and some of them also CD8 response. The target in most of them is the spike, but the construct that people are using are, there is a benefit of a lot of science in the MERS and SARS, and the science is to produce the protein in the right conformation, which is the pre-fusion form of the protein. We had the same problem with RSV vaccines. RSV vaccine failed for years because we were using the most stable form of the protein, that is the post-fusion of the F protein of RSV. Now that we have a pre-fusion, stabilizing the pre-fusion conformation, we have vaccines in the pipeline that are very, very effective in inducing neutralizing antibodies. I think that for COVID is the same. We have the right antigen, the vaccine may work, but to prove that it works, to exclude that there is disease enhancement, it takes a long time. And also to scale up manufacturing takes a very long time. So probably end of next year is the best we can think about. End of next year, we will have still a third season, a second season, and then a third season of COVID. I don't know. I'm not an epidemiologist. I hope we will not, actually. I hope I addressed your question, and I'm sorry to be a little bit too long.
No, not at all. I mean, I suppose one other thing that many of us are wondering about is, especially as it is unclear, and I should just say that it's not. I'm not sure if everyone can see the outcome of the voting, or if it's only the organizers at the moment. The voting is standing at 2-1. That you think that we won't have a vaccine anytime soon that will help us to get out of the epidemic. So currently you've voted 2-1 as pessimists. The glass is mostly empty.
Well, for me, sorry, I just want to add one thing. The vaccine is going to be an insurance in the in the scenario that is the worst case scenario. The worst case scenario is only very few people have been exposed. The serological assay in Italy, and Italy was actually had a lot of cases, but still, when you run serological assay on the broad population, it's less than 5%. So doesn't mean that people have not been exposed, but the people that have been infected is really a very, very minor percentage of population. And if everyone or the majority of people are susceptible to the virus, and I understand this was not the hypothesis previous talk, and it may still be, there is a chance that the virus will continue to circulate for years and pops up again every season until we will have a vaccine that can generate a robust herd immunity. And also, this hypothesis states that having herd immunity without lockdown through free distribution of the virus will be very, very painful in terms of number of deaths. Now, that's the theory behind the vaccine, is that doesn't matter if it comes in 2 years, it will still be useful to generate a robust herd immunity, more than 60-70% of the people with high neutralizing antibody titers. That's the hypothesis for me that support the investment in vaccines.
And then just then before we move on, one other question that's come up is, could you say anything in parallel about development of other types of therapeutics, antivirals, small molecules? I mean, I guess as well as the great capacity for vaccine development, Big Pharma also has a fantastic capacity for small molecule-based drugs.
So for other therapeutic agents, the advantage is that you have to make the doses targeted to people that are sick, right? So it's a lot easier to scale up manufacturing for things that has to be given to COVID positive, seriously. I mean, people with disease are not so many. So there is not the same challenge that we have with vaccines, where if you want to create herd immunity in the world, you need billions of doses. Here, for therapeutics, you need thousands of doses. So it's a completely different challenge. The other advantage of therapeutics is that you can test if they work or not directly on people that are sick. So monoclonal antibody, neutralizing monoclonal antibodies, I believe, will come before the vaccine because it will be very easy to assess if they work or not, especially in the early phase of disease. You give them monoclonal and you see in a randomized clinical trial how many people progress to intensive care, how many will actually have only mild disease. So for small molecules, generally it takes more to find specific antivirals. Maybe I have a lot of faith actually in as a first response intervention on monoclonal antibodies. Actually. I think that will help to manage the situation of people that are sick before we will actually prove that the vaccine is protective on very, very large clinical trials.
Thank you very much, Ennio. That was great. I'd like to bring in Ishaan Gupta now from the Indian Institute of Technology. Ishaan, are you there?
Yeah, I'm here.
Hello.
Share my screen. I can see, so I'm much more recent than Ennio. I was there from 2010, 2015. I think and I overlap with Janosch, Federico, and Sergio, actually. A couple of other people I missed. Hi to you. So I'm going to talk to you guys about 3 points. One about the science that we've been doing, and 2 about the nature of science around COVID-19. So let me just figure out how to share my screen, because I have 2 screens. Is my screen visible?
Yes.
I guess. Okay.
Yes, we can see it.
Great. Okay. So I think this is a great time to do science, and I mean this pandemic has actually shown us this. So it's I think we're in the zone of peer-to-peer science, which is not just preprints but also exchange of protocols and materials. And here, I think last time there was Vicente Pellicano presenting the protocols that they have developed. So we had a big problem in India, which we're still facing currently, is the shortage of cold supply chains as the movement has stopped because most of the enzymes we are basically dependent from outside companies for that. And just there are scientists who are out there. You don't have to reinvent the wheel, and they are willing to help. And so you should definitely reach out in such a case. And I reached out to both EMBL alumni and other people who are doing similar job of sharing their protocols and science. Secondly, I think everybody who has been following the research around COVID would be essentially reading the research of bioRxiv or medRxiv before it hits any of these big journals. So I think although there's a bit of noise there as well, but I think this is really a testament to how science can be done in a fast and accessible way. So it's one for peer-to-peer science, right? So now coming to the topic at hand. So basically, after EMBL, I was in the genome biology unit with Lars Steinmetz. After that, I did 2 postdocs, one in human genetics and another one in single-cell biology, single-cell gene technology development. And the work that I'm going to present is something around that. So we have 2 questions, to put mildly. How does COVID kill? And particularly which parts of the body are actually affected by the infection? So lungs is an obvious part. But particularly, if you talk to the clinicians, what they're seeing is a lot of coagulation. This phenotype is like majorly seen across all sites. It was reported both in Italy, China, and doctors here in India are also seeing it in a large scale. So the whole question that we wanted to answer is what parts of the body are infected and what cells in that body in the body are infected. So typically, when you are dissecting the molecular biology of COVID, people have taken 2 approaches. One is the approach of actually measuring, quantifying the change. This change can be quantified through histopathology, through actual measurements of molecular biology, through the transcriptome and the proteome. And you see that unlike other COVID viruses, SARS-CoV COVID-2 has some interesting manifestations in the host reaction to the virus. And but these are just giving you some sort of overall picture of what's happening inside the body, but not exactly the geography of the virus. So like histopathology gives you like for a few genes, but we want to basically utilize these new technologies to know more about exactly where the virus is affecting. The second way of understanding how the virus goes about its infection is by actually looking at where in the body the virus is interacting. So everybody's talking about the spike protein. It's nice, spike protein, TMPRS2. But the virus has like three parts to its life cycle, right? The virus has to enter the cell, the host cell. It has to do something inside the cell, process, and it has to exit the cell. And during all these processes, the virus itself or the viral proteins themselves have to interact with the host. So there were two studies which recently which published like early late March, where they actually use this affinity purification mass spectrometry to take a viral bait protein, which is shown here in a diamond, and see which all human proteins they interact with. Although two studies don't overlap very well, they have similar tags on their bait proteins. You get a picture of exactly what all different kinds of human proteins the virus is interacting. So moving ahead, what we did was you have all this data which describes exactly what is the molecular phenotype or how does each cell in each organ looks like. So this has been made possible by this technology called single cell gene expression. And I can talk more about it. But the basic idea is that now we know we have a map of exactly which genes are expressed in which cells of the body. And using this, we basically hypothesized two things. First, that gene expression changes that you would be seeing. So gene expression changes at the level of the protein or the transcriptome in COVID-19 patients will be specific to only a few cell types in the affected organ. And secondly, these cell types that will be affected in the organ should actually also highly express the genes that are interacting with the virus. So for the sake of our initial study, we chose the lungs, which data is available. We chose the esophagus, which is the food pipe, which is actually very similar to in structure with the air pathway. The spleen and PBMCs, this is the peripheral blood system, because you see a lot of hemodynamic changes. Hemodynamic would basically mean that there's a lot of changes at the level of your blood. So long story short, this is what we found. So when we deconvolve, so deconvolution essentially is the idea where you have an organ, like an organ is made up of different cells. Similarly, the gene expression that you have in that organ is made up of expression of these different cells. The change in the expression of the organ would also be a combination of the change of expression in specific cells. That's the hypothesis. And when we actually apply this deconvolution method, what we find is that across lungs, across these different tissues, you have monocytes and cells that are belonging to the vasculature. So you have blood vessels or you have the epiblast cells. So the monocytes are going up and the vasculature is going down. So the signature for this vasculature is going down. When you take a look at data from COVID-19 patients versus healthy patients. If you go to look at the interactome, so which cells are expressing the human proteins that are most interacting with the virus, there also you see that some of these cells are coming back again, like monocytes are coming back up, blood vessels are coming back up. But we find that in spleen and PBMCs, actually, we find enrichment for platelets, which is interesting because there is no data out there which actually suggests that COVID-19 could infect platelets. There's only one old 1980s paper which shows that the virus can actually get inside the platelets. So that's up for debate. But this entire exercise, so I'm just shortening it up, shows that in case of clinical science, there is something called Vircho's triad. I'm probably not pronouncing it correctly, which is getting reinforced in COVID-19, which leads to this coagulopathy phenotypes through upregulation of monocytes, which are affecting the blood vessels and platelets. And interestingly, we find that the platelets in older patients are actually primed more towards platelets from patients that are severely affected. So that's the story in brief. I think it's too much of a shortening, so you can ask me more questions at the end. And last, I would like to end with what are we? I like this photo quite a lot, because right now we are facing the first pandemic, I would say, in a series of pandemics that might be associated with climate change and what's coming. So I would really, maybe this would be a good forum to discuss, and maybe the EMBL Alumni Association can also take a lead in this. I think there is this dire need to start an open source biosurveillance platform which is not affected by the politics of the different countries, and basically leading to decentralization of protocols, materials, and research. I think we really, I do not feel like spending $2,000 on making my science known in an article when I can actually get it published. Not published, but like get it on a platform like a medRxiv or bioRxiv, and it actually reaches the people, and there's an open discussion. As Michael was saying initially, that Twitter is like a very good platform for that. So I mean, they really tell you if you've done something wrong. They have no qualms about shitting on your doorstep to tell you that you've done something wrong. So yeah.
Okay, thank you very much, Ishaan. Now, you've raised a whole bunch of different topics there, and I think we can open up for questions. I mean, one obvious question that comes up is: do any of your data, particularly Interactome data, suggest any promising additional candidates, following on from Ennio's talk, for antiviral therapies that would open up some new weak point in the virus that could be targeted?
So we do actually. There is an enzyme called granazyme A that interacts, that basically is released by the monocytes and interacts with the virus, with the platelets, to enhance this triad. So to enhance the coagulability phenotype, and it's actually highly expressed in old patients as well. So that's a potential target, which is an enzyme. So you can probably make blockers and all that for that, and basically test it as well in the system. So yeah,
okay,
that's one clear target that's out there.
So Ishaan also raised the point that we should be addressing this at the global level. And for those, I know some people have joined the meeting after we started today, and so I just want to come back to the point right at the beginning that when we wished everyone Happy World Environment Day, because this United Nations have set up June the 5th since 1974 as World Environment Day to draw attention to global challenges around the world, including loss of biodiversity, which is a theme today, but also challenges like the spread of global infections. And I think it raises a lot of issues that are relevant to this organization. I mean, science is absolutely I think one of the ultimate global activities. I've always felt incredibly proud and enjoyed the fact that we're part of a global community. And I think it's great to see again that at these Coffee with EMBL sessions, we have so many people from countries and continents all around the world who interact with each other. This is not a political forum per se, but I think it would be wrong if we didn't make some mention today. If I could, on behalf of the association, share our common disappointment and frustration and anger, I suppose, at the events we've seen unfold over the last week in the US. The science community is one, and I'm proud to say that while no organization or community is by any means perfect, I've always been very pleased at how little partisanship or racism has been shown. And it is, of course, very disappointing that in year 2020, we're still faced with some of these issues. But we have to, keep looking on the positive side of things, but use these as opportunities to highlight what we can do better in future. I strongly feel we should recognize problems in the past. We can't change the past, but we can learn from it and try to do things better in the future. And Ishaan certainly highlighted some of the things that we can do. And I know I've done this myself in my career, and distribute reagents from my lab to anyone who wants them from anything we've ever published, and I very much support also his viewpoint that the more we can pool resources and share them, the more we can join together to rise to some of these global challenges. So thanks to everyone who's spoken today. I'd also like to just open up, if anyone who's attending the meeting would like to add a comment. We're very happy to hear from anyone who'd like to contribute. Francis Stewart, are you there? Can I see Francis?
Hi, Angus.
Can we?
Marvelous.
Hi, how are you doing? You've got the floor.
Yeah. So I'd like to ask Michael. It seems like he is proposing or contending that the declaration of the pandemic was an overreaction. Mm-hmm. And if so, how has this overreaction arisen?
Obviously, I don't know, but as I said, I was following things from China in the end of January. It sort of ended. We came into March, and things seemed to be going okay. I felt that I was very, very concerned about what was happening in Italy, and we were looking at the numbers, and as soon as the Italian cases stopped increasing day by day, we were very encouraged. So there were signs that everything was still fine, and then suddenly, I almost feel that the leaders panicked, and then everyone else panicked. The situation in the United States is actually a little different. I don't know if you know, there was this case just recently where a paper showing that chloroquine is ineffective was found to be fraudulent.
Yeah.
It had been fast-tracked through Lancet. It had been fast-tracked through peer review.
Yeah.
I'm a very apolitical person. I'm so apolitical that when I watch sports, which I like doing, I don't actually care which side wins. I like the moves. And it seems that in this case, there is so much political antipathy, particularly in the United States, that science and truth don't matter anymore. And I think that's a big, big mistake. So I think that, and the fault isn't just on one side, it's everywhere. So I don't know, but I think that there's a lot of politics. I mean, the shorthand is the leaders panicked, and then the rest of the world panicked. Sometimes I think...
So maybe I can ask then, your analysis seems to imply that inevitably the infection rate expires as a natural consequence of the parameters of this infection. However, if you look at what has happened around the world, the governments who have behaved carefully and quickly, like Australia, like New Zealand, like Germany, have managed to put a lid on this whole thing, whereas the governments who have made a mess of this, like the UK and the United States, and I don't know if you've seen the incident rate in Texas is going up. It's lifting off again. Your explanation about the natural truncation of the infection path does not appear to fit with the evidence of the political responses.
Let me explain. It's absolutely clear that if one city is infected and all transportation from that city to other cities is curtailed, the infection will not spread. So if you take a country if you compare Switzerland to Austria, two countries that are very similar geographically, very similar latitudes. Switzerland had 2,000 deaths. Austria had you could argue that Austria has managed to put a lid on it, but failing a vaccine, Austria now has 1,800 people who are going to be very vulnerable. So I don't know whether it's also turned out that Switzerland, with this 2,000 deaths, has zero excess mortality. Those deaths just fell into the hole created by the absence of flu. So I think that you can stop it spreading. Whether that's a good idea or not, I don't know. What seems to be clear now is that Sweden is not going to have tens of thousands of deaths. Sweden is definitely trying to spread the disease. If you look at the curve of cases increasing, it's not natural. Cases are increasing completely linearly. Somebody is controlling it. But it's not, so you can definitely, if you're an island and you don't let anyone come to your island, you will not have corona. But is that the right thing to do? I don't know. All the epidemiologists that I read about before this happened said that closing down borders is not a good idea, because essentially it's like keeping a lot of dry wood, and at some point a spark is going to get to it. I think a large fraction of the world has not reached anything like a saturation level. When we were hearing about the talk on Italy, southern Italy definitely could be an area. I think that, I think also when you think about this, you have to realize if lockdown caused no damage whatsoever, no economic damage, no social damage, no other damage at all, no political damage, I think it would be a great thing to do. I think for a lot of academics, the greatest holiday in the world. They didn't have to teach. They basically could do their research. But for people who are not so lucky, this has been a disaster. And at some point, we have to look at the excess death, the excess money caused by a disease that has basically had a burden of death that is not dissimilar from flu. Whether that burden of death would have happened without the lockdown, we don't know. We can look at Sweden, we can look at Switzerland, both countries that had very, very mild lockdown. I'm not saying there shouldn't be social distancing. I think wearing masks might have been the 10-cent solution that saved $100 billion. It's just that we had to be clever. We had to use smart lockdown. The lockdown that we used is coming from a medieval playbook. You don't leave your house. And what was interesting was, while this lockdown was going down, I was in Israel, very strict lockdown. They had better numbers than Austria, better numbers than Australia. They were very proud. They used to have a lot of people who were potentially vulnerable. But what was interesting was when I went to the store for my one visit today buy food, the storekeeper who spoke to me did so unprotected. He was spraying his virus on me. I'm sure he wasn't ill, but people are not feeling ill for this disease. So I think a little bit of thought about what leads to the epidemic spreading would have been a really, really good idea. And again, we have the example from Japan, Taiwan, China, Singapore, Hong Kong, where this was, and South Korea, where this was controlled because people there wear face masks, not to protect themselves, but to protect everybody else. But it's going after very selfish, self-centered Western cultures. I also think that there's a very strong correlation between the death rate and income inequality. Because this is what's happening in the United States. When I talked about my death rate numbers, that was for Europe. In the United States, the death rate for people under 65 is more like 35% instead of 8%. Just think what that means. A very unhealthy population, no health insurance, and this is a problem. So I think we have to think what could have been done. If the United States had spent one fraction of the money it spent on lockdown, were lost on lockdown, increasing longevity, we could have saved millions of people. There was this huge panic. The media counted every death. And I am not prepared to accept that the death of a soldier aged 20 is the same as a centurion dying. When you get old, you die, and that's the way life is. And because you die when you get old, we have young people. So I don't believe we can move away from that. If we want to count every death, if you want to call me a granny killer, please go ahead. I am 73 years old. My mother, who is locked down in London, is 105. We're not worrying about that. The fact is, I have had 73 years of wonderful life. And we are killing young people, especially disadvantaged young people, because we're spending money on old people, and that money should be going to young people. The real revolution here is going to be the baby boomers seeing their sunset years and not controlling everything.
Well, that's remarkably well put.
Very nice.
Thank you.
Yeah, no, it is very well put. And again, I really would like to offer the floor for anyone who would like to speak. One other comment, perhaps, to bring this along is I have to say one of the concerns I have, Michael, is that as serious and problematic as the COVID-19 virus is, it could still be a lot worse. There have been viruses recently seen, including Nipah virus, Ebola virus, And while we can argue about the trajectories of infection and whether they could spread, I think we have to be very concerned seeing how our different societies reacted to this spread. I think we should count ourselves very lucky that as serious as it isn't much worse, that the mortality rates are not even worse than they are. And I think we shouldn't assume that there won't be a future pandemic, which could happen any time, with a virus that is capable of killing a lot more young people. You raised this very relevant point about when a virus contributes to terminating the life of someone. How many years of life expectancy did they have left? In the 1918 flu epidemic, many young people were killed by the virus at the prime of their life. And that could happen again. So what lessons do you think we've learned from this? That when the next epidemic emerges, whether it's from Asia, Africa, Europe, or anywhere else, how should we react instead? I wonder if you could maybe start.
I have a clear answer to this. Firstly, I don't think you have to invoke viruses. We all know that in the next 20 or 30 years, there are going to be massive challenges that arise from global warming. And, if we look at this as being a dress rehearsal for how we deal globally with problems, I think there is no doubt in my mind that the whole world has really got a very solid F grade on this. But when I was thinking about this, and I also am very involved with computing and machine learning and things like that, I was actually very encouraged by the fact that machine learning is getting better and better. Artificial intelligence is progressing, and exactly the same way that even those of us who thought we had great navigational skills wouldn't think about getting into a car without turning on a GPS driven by Google or Waze or whatever. I really, really hope that in the next few years, we will learn to go to our phones, to say, Alexa, Google, and the third one that I cannot say right now because they will respond to me, should I panic? And listen to the answer and believe it. Because it's clear to me that conventional media has been an incredible failure. All they wanted to do was maximize advertising revenues by making you listen to the daily count of death. They never, ever once said what people normally die of. I, for example, I'm a subscriber to the New York Times. I think they front 100,000 people who died of coronavirus, many of whom are not excess deaths. The excess deaths in America are much more like half of that. But in doing so, they put the names on the front page of, let's say, 100,000 people who died in the same period in the United States, another 600,000 people died. So why do the ones who get labeled with corona become heroes? These are not soldiers. People die. And maybe newspapers should every day record 150,000 people die every day in the world. Almost 2,000 people die every day in Italy. In countries, people die. Maybe the day, and these people are dear people, their grandparents, their parents. Some of them are young people. We never, ever in the past glorified passing away of people like this. If we had, maybe it would have been a good thing. There would never be any wars. Because in wars, the loss of life is colossal. So I think that, I don't know why it was done. I find it crazy. But, we also see certain institutions that we have as being really bad ideas. Nursing homes have probably been responsible for half of the death globally. And these are people who have basically been, not being taken in by their families. Maybe they don't have too many children. And being left basically in a typical nursing home in America without coronavirus, 1/3 of the inmates die every year. That's a very, very high death rate. So I think we have many, many things that are going on here. But I think we need to start being smart. In much of the world, we have leaders that none of us would employ in our labs. We have leaders who are, imagine you chose a professor by a beauty contest. Would you find an airplane where the pilot wasn't a really good pilot, but boy, was he popular. He had more likes than any other pilot. Or maybe a brain surgeon who hadn't been trained, didn't really care, but he was popular. This is how we're appointing politicians. It is a disgrace. The only countries that don't appoint politicians this way are China. I'm not saying I agree with the Chinese system, but people in China or go through a very, very strict meritocracy. And these people can actually do something. They're not just chosen, they're not popular. And this is a huge mistake. It's fine for a world where there are no crises. Likes are great, but the real world has problems. And if we go back and look at the politicians we have now in the Western world, so what we had 50 years ago, we have nothing. So it's a real disaster. And I think we need to just hope that from very soon on, maybe you and I will not be asking our computer what to do, but there'll be a law saying that every single politician needs to at least ask his computer what the computer would do, and that gets recorded.
I'm sure you're not suggesting that if Donald Trump applied to come and work in your lab, you wouldn't accept him, because he would clearly be the best co-worker you've ever had. I'm sure he would say that.
It would help my grants no end.
So one of the things, Pat, just to follow up on that is, before we started today, you and I both shared our frustration at the relative toothlessness and lack of effectiveness of some of the world's leading academic institutions at presenting a more coherent policy towards government and the media, or their ineffectiveness. How do we do better? We can complain and we can wring our hands and argue that the National Academy isn't doing enough, or the Royal Society or whoever isn't doing enough, and the wrong people are advising government, and the media are listening to the wrong people. How do we as a community do better? Because, we are already a strong enough community on these calls that I think we could do something positive. And I'd like to see one outcome of this being, I actually agree with this and everything you've said today. I think criticism of what's happened is an important point, but I think it's also really useful if we could try to channel our energy into thinking of something constructive we can do. And I hope if you have got an idea, Michael, or if anyone else would like to contribute to that, how can we effectively contribute towards informing the public respectfully of concerns and fears, but to provide a more balanced, accurate view of the reality of the situation, and to make sure that that's communicated also to governments?
If you're asking me, I was very adverse to social media after going to Stockholm, because I was just so exposed in. If you Google my name, there's a huge number of people. My web server went down. I didn't really care. Who needs a web server? I think if this were to happen again, I would make sure that I had access to my own web pages. I didn't have to post my reports on GitHub because there was no one that would report them. I think scientists have a number of obligations. One of them is to answer emails. Scientists in Britain do not answer emails. It's kind of amazing. I emailed all the epidemiologists, University College, Imperial College, Oxford. Oxford answered me a month later. I got one reply from Imperial College only when I brought in the head of the Royal Society, who's a friend. So this kind of lack of communication is awful. What you're doing here is exactly the right thing to do. I think that, as I said, I was very scared of Twitter. The Twitter-in-chief of the world is not somebody who I really would like to emulate. But on Twitter, I have really been amazed by the quality of information, plus the documents I can get. I have been given documents on all kinds of things, hearing why the Italians made their decisions and things like this. Tons of things in Swedish come my way. So I think that, I sort of see, and I also must say that taking the point that was raised by Ishaan about preprint servers, my very first report that I wrote on the 2nd of February, I tried to get into Biox Archive, Medics Archive, and ArchiveX, and they all said, since I didn't have an introduction, conclusion, and references, it could not be archived. Now, I don't mind. That's fine. It's a rule. But it's not really a very smart rule, because basically there was very important information in there that could have gone on display. I just gave up. We're now about to write a very important paper, which is actually worth publishing. Those things were not really worth publishing, and that will definitely go to a server. But I think that there's a lot of gatekeepers in our society. We don't realize them. Gatekeepers who are editors of newspapers, gatekeepers who are running programs. what's been amazing to me is the number of podcasts. People can become their own studio by being proactive. So this is a very important medium. We have a lot of gatekeepers in privileged positions controlling things. And it's very frustrating because I try to send an op-ed to the New York Times. I didn't even get an answer. The next week, one of the editors in the New York Times referred to me. They contacted me to see that I was me. That was it. So I think, another thing is I actually sent the head of EMBL, asked everyone to send them information what they were doing on COVID. I sent her all my reports. I'm quite sure she never had seen them because people don't understand how to deal with email. Email has to be triaged. You look for attachments. You do it in a smart way. We're in trouble. We really are in trouble.
Thank you. I would just like to also thank you as well as the other speakers today. I've once again really enjoyed the session. I think it's raised, I've learned a lot. It's raised a lot of really important issues I want to go and think about. And it's made me even more convinced that we have a forum here that is incredibly useful and a vehicle we can build on in future to address a number of issues going forward.